Hello everyone, today we are going to learn Chapter 2 Exploring Substances Acidic, Basic and Neutral from your science textbook. In this video, I'll be covering each and every line from the chapter so you don't miss anything important. Make sure to keep your notebook and pen ready and take notes while watching. This will help you remember the concepts better. You can pause or rewind any time to write things down. So, let's begin. On 20th February, the school hosted a science fair to celebrate National Science Day. At the entry gate, siblings Ashwin and Kirti were greeted with a white sheet of paper. They were curious to know why a white sheet of paper was given to them. A few steps ahead, there was a volunteer spraying a liquid on these sheets of paper. The siblings also got their white sheets sprayed. To their surprise, the words "Welcome to the wonderful world of science" appeared on the papers the moment the liquid was sprayed. Figure 2.1. They were excited and eager to know how this happened and the reason behind it. Their curiosity was partly satisfied at the Colourful World of Substances stall. They saw many activities showing color changes on mixing different substances. They decided to explore these changes further. Let us join them on this learning adventure. So, every year India celebrates National Science Day on 20th February to honor the discovery of the Raman effect by the Indian scientist Sir C.V. Raman. On this day, schools and colleges often organize science exhibitions, fairs and activities to encourage students to develop an interest in science. In the chapter, Ashwin and Kirti's school also organized a science fair to celebrate National Science Day. As the two siblings entered the fair, they were given a plain white sheet of paper at the entrance. Now imagine being handed a completely blank sheet when entering a science fair. Most of us would immediately wonder why we were given it. The Ashwin and Kirti felt exactly the same. As they walked further inside, they noticed a volunteer spraying a liquid on the sheets of paper. Curious to see what would happen, they also got their sheets sprayed. Suddenly, something amazing happened. The blank paper was no longer blank. The words "Welcome to the wonderful world of science" appeared on it as soon as the liquid touched the paper. What seemed like an ordinary white sheet was actually hiding a message. Naturally, the siblings were surprised, excited and eager to know the scientific reason behind this interesting event. And they reached a stall called Colourful World of Substances. Here they saw several activities in which substances changed color when mixed with other substances. These color changes immediately caught their attention. They began to think that perhaps the mystery of the hidden message on the paper was also connected to a color change. Their curiosity was now partly satisfied because they had found a clue, but they still wanted to learn more. So, just like Ashwin and Kirti, we are now going to explore how certain substances can cause color changes and what these changes tell us about the substances. So, in this chapter, we will discover how scientists identify the substances as acidic, basic or neutral. 2.1 Nature, our science laboratory. 2.1.1 Litmus as an indicator. Litmus is a natural indicator which helps us identify whether a substance is acidic or basic by changing its color. Because of this property, litmus is commonly used in science experiments to test different substances. Activity 2.1 Let us explore. Collect samples of lemon juice, soap solution, amla juice, tamarind water, vinegar, baking soda solution, lime water, tap water, washing powder solution, sugar solution and salt solution. Take a strip of blue litmus paper and cut it into small pieces. Spread these pieces on a clean and dry white tile. Using a dropper, put one drop of each of the samples one by one on these litmus paper pieces as shown in figure 2.2a. So, this is an activity using blue litmus paper. We will collect different samples such as lemon juice, amla juice, tamarind water, vinegar, soap solution, baking soda solution, lime water, tap water, washing powder solution, sugar solution and salt solution. How to prepare lime water? Do not confuse lime water with the word lime, which is a fruit similar to lemon. Lime water, that is solution of calcium hydroxide in water, can be easily prepared by mixing lime that is tuna, that is calcium oxide in water and leaving it undisturbed for some time, say an hour. Filter the liquid into another container and use it as lime water. So, the lime water used in science is not made from the lime fruit, which is similar to a lemon. It is a solution of calcium hydroxide in water. To prepare lime water, lime, that is tuna, is mixed with water and left undisturbed for about an hour. During this time, the undissolved particles settle at the bottom and the liquid is then filtered into another container. The clear liquid obtained after filtration is called lime water. This lime water is used in science activities and experiments. Coming back to our experiment. First, take a strip of blue litmus paper and cut it into small pieces. Place these pieces on a clean and dry white tile. Then, using a dropper, put one drop of each sample on separate pieces of litmus paper. Do you observe any change in the color of the blue litmus pieces? Record your observations in table 2.1. Repeat the same activity with pieces of red litmus paper as shown in figure 2.2b and record your observations in table 2.1. So, after placing the samples on the blue litmus paper pieces, carefully observe whether the color of any piece changes. If the blue litmus paper turns red, it indicates that the substance is acidic. Record all your observations in table 2.1. Next, repeat the same activity using red litmus paper. Place drops of each sample on separate pieces of red litmus paper and observe the changes. If the red litmus paper turns blue, it indicates that the substance is basic. Again, record your observations in table 2.1. Now, let us analyze table 2.1 and sort the samples into three groups as follows. Group A with samples that turn the blue litmus paper to red. Group B with samples that turn the red litmus paper to blue. Group C with samples that do not affect either of the two litmus papers. Record the data in table 2.2. So, we have completed the litmus test and recorded our observations in table 2.1. Now, instead of looking at each sample separately, let's organize them into three groups based on how they affect litmus paper. This will make it easier to identify patterns. First, collect all the samples that change blue litmus paper to red and place them in group A. Next, put all the samples that change red litmus paper to blue in group B. Finally, place the samples that did not cause any change in either blue or red litmus paper in group C. By sorting the samples in this way, we can easily compare their behavior. Notice that the substances within each group show a similar effect on litmus paper. This suggests that substances in the same group may have something in common. In the next part, we will explore what makes these groups different from one another and what these color changes actually tell us about the substances. I am curious to know what are these red and blue litmus paper strips made of. Why do they change color when drops of some samples are put on them? So, we have just seen something quite interesting. Some substances change the color of litmus paper while others did not. This naturally makes us curious. How can a simple piece of paper change color? What is special about these red and blue litmus papers? Are they made from some unique material? And why do only certain substances cause a color change? Let's find out the science behind this fascinating observation. Let us find out. Litmus is a natural substance obtained from lichens. It is available both as a solution and in the form of paper strips known as litmus paper. The litmus paper is available in two colors, blue and red, as shown in figure 2.3. Substances that turn blue litmus paper to red are acidic in nature, while those that turn red litmus paper to blue are basic in nature. Since litmus shows different colors in acidic and basic solutions, it is called an acid-base indicator. Some other substances, both natural and synthetic, can also be used as indicators. Synthetic indicators are made in laboratories and you will learn more about them in higher grades. So, litmus is a natural substance obtained from lichens. Lichens are special living organisms made up of an alga and a fungus living together. They help each other and grow as a single organism. Lichens are commonly found on rocks, tree trunks, walls and other surfaces. Scientists obtain litmus from these lichens. And it is available in two forms, as a solution and as paper strips called litmus paper. These litmus papers come in two colors, blue and red. Substances that turn blue litmus paper red are called acidic substances, while substances that turn red litmus paper blue are called basic substances. Because litmus helps us identify acids and bases through color changes, it is known as an acid-base indicator. And litmus is not the only indicator. There are many other indicators that can also show color changes. Some are natural like litmus, while others are synthetic, meaning they are made in laboratories. You will learn more about these indicators in higher classes. Holistic lens. Lichens are formed by the association of two living organisms, a fungus and an alga. They grow on rocks and trees in regions that have abundant rainfall and clean air. Do you find lichens on trees in your neighborhood? So, as I told you earlier, lichens are formed when two living organisms, a fungus and an alga, live together and grow as a single organism. They are usually found on rocks and tree trunks in places that receive plenty of rainfall and have clean, unpolluted air. This is why lichens are often considered indicators of clean air. If the air is heavily polluted, lichens usually do not grow well. Now, let us classify the substances sorted in Group A, Group B and Group C in Table 2.2. The substances in Group A such as lemon juice, amla juice, tamarind water and vinegar turn the blue litmus paper to red, implying that these substances are acidic in nature. The substances in Group B such as soap solution, baking soda solution, lime water and washing powder solution turn the red litmus paper to blue. Hence, these substances are basic in nature. The substances in Group C such as tap water, sugar solution and salt solution did not change the color of either litmus paper. Can you predict their nature? These substances are said to be neutral because they are neither acidic nor basic. So, after grouping the substances based on their effect on litmus paper, we can now understand what these groups actually represent. Group A contains lemon juice, amla juice, tamarind water and vinegar. All these substances change blue litmus paper to red. This common behavior tells us that they belong to the same category. Such substances are called acidic substances. Group B has soap solution, baking soda solution, lime water and washing powder solution. These substances change red litmus paper to blue. Since they show a similar effect on litmus paper, they are placed in another category called basic substances. Group C contains tap water, sugar solution and salt solution. These substances did not change the color of either blue or red litmus paper. Since these substances are neither acidic like Group A nor basic like Group B, they are called neutral substances. In other words, neutral substances are neither acidic nor basic. Activity 2.2, let us relate and explore. Are all the substances in Group A of Table 2.2 edible? Have you ever tasted these edible substances? Can you recall their taste? You will find that all these substances taste sour. Thus, we can say that substances that taste sour tend to contain acids and are acidic in nature. Caution: do not taste anything until asked to do so. Do not taste any unknown substance. So, let's look again at the substances in Group A: lemon juice, amla juice, tamarind water and vinegar. Most of these are edible substances that we use in our daily lives. Have you ever tasted any of them? If you recall their taste, you will notice that all of them taste sour. This observation gives us an important clue. Substances that taste sour generally contain acids and are therefore acidic in nature. So, sour taste is one of the common characteristics of acidic substances. Also, remember that we should never taste an unknown substance to find out whether it is acidic or not. Scientists use safe methods and indicators such as litmus paper to test substances. Therefore, do not taste anything unless you are specifically told it is safe to do. Some common edible substances and the names of the most common acids present in them are given in Figure 2.4. Figure 2.4, the most common acids present in some edible substances. Here we can see that orange contains citric acid, And amla contains ascorbic acid, that is vitamin C and citric acid. And mango contains tartaric acid. And tomato contains citric acid and oxalic acid. So, different edible substances contain different types of acids that give them their characteristic taste. So, even though all these substances are acidic, they do not contain the same acid. Different foods can contain different acids, but they all show acidic properties. Find out and write the names of the most common acids present in the following substances. Lemon contains citric acid, which gives it a sour taste. Curd contains lactic acid formed during the fermentation of milk. Tamarind contains tartaric acid, while vinegar contains acetic acid. Although these foods contain different acids, they all show acidic properties. Now, let us take one of the substances, baking soda solution from group B. Rub the baking soda solution between your fingers. What do you observe? It feels soapy or slippery. Basic substances are generally slippery to touch. Also, bases generally taste bitter, but everything that tastes bitter may not contain a base. For example, bitter gourd, that is Karela, possesses a bitter taste, but it is not basic in nature. So, now let's look at the substances in group B. Take baking soda solution as an example and rub it gently between your fingers. What do you notice? It feels soapy or slippery. This gives us an important clue about basic substances. In general, basic substances are slippery to touch. Another common property of bases is that they usually taste bitter. However, we must be careful here. Just because something tastes bitter does not necessarily mean it is a base. For example, bitter gourd, that is Karela, has a bitter taste, but it is not basic in nature. Now, I can check the nature of floor cleaning liquid using an indicator. If litmus is not available, or there are some other natural substances that can serve as acid-base indicators. So, we have learned that litmus can help us identify acidic and basic substances. But there are also some other natural substances around us that can act as acid-base indicators just like litmus. So, let's find out. Red rose as an indicator. You might have observed many colored flowers in your surroundings. Try making your indicators using these flowers. Let us prepare. Collect some fallen petals of red roses available in your surroundings. Figure 2.5. It is advised not to pluck flowers. You may pick petals or flowers fallen on the ground. Take a fistful of the collected petals of red roses and wash them with water. Crush the petals using a mortar and pestle. Place them in a glass tumbler. Pour some hot water into the glass tumbler to ensure that the crushed flower petals are completely immersed. Caution: perform this step under the supervision of an adult. So, we just asked whether there are natural substances that can act as indicators besides litmus. To find the answer, let's explore a common flower that many of us have seen, the red rose. Surprisingly, the petals of a red rose can be used to prepare a natural indicator. In this activity, we will make our own indicator using red rose petals. First, collect some fallen red rose petals from the ground. We use fallen petals so that we do not harm the plants by plucking flowers. Wash the petals and crush them using a mortar and pestle. Then place the crushed petals in a glass tumbler and pour some hot water into it so that the petals are completely immersed. The hot water helps extract the colored substance from the petals. So, you should be careful while handling hot water and perform this activity only under the supervision of an adult. Cover the glass tumbler with a lid. Wait for 5 to 10 minutes till the water becomes colored, figure 2.6, and filter it. The filtrate, liquid after filtration, is the required flower extract, figure 2.7, to be used as an acid-base indicator. So, after pouring hot water into the tumbler, cover it with a lid and wait for about 5 to 10 minutes. During this time, the colored substance present in the rose petals slowly mixes with the water, causing the water to become colored. Once the water has become colored, filter the mixture to separate the liquid from the crushed petals. The liquid that passes through the filter is called the filtrate. This filtrate is actually the red rose extract that we wanted to prepare. This flower extract will now be used as natural acid base indicator, and in the next activity we will test different substances with this extract and observe whether it changes color. Figure 2.7, test tube containing the red rose extract. So, this is a test tube stand and it has a test tube with red rose extract. Activity 2.4, let us find out. Place 10 to 20 drops of the prepared red rose extract in each of the two small transparent bottles or test tubes. Mark them A and B. So now you need to take two small transparent bottles or test tubes and place 10 to 20 drops of the red rose extract into each one. To avoid confusion during the experiment, label one test tube as A and the other as B. Add 20 to 30 drops of lemon juice in test tube A and 20 to 30 drops of soap solution in test tube B with the help of droppers. Observe and record any color changes, figure 2.8, to the extract in table 2.3. Repeat the same with the other samples used in activity 2.1 and record your observations in table 2.3. So now we need to add 20 to 30 drops of lemon juice to test tube A and 20 to 30 drops of soap solution to test tube B using droppers. Then observe the color of the rose extract carefully. Does it remain the same or does it change? Record your observations in table 2.3, and after this repeat the same procedure with other samples used earlier such as amla juice, tamarind water, vinegar, baking soda solution, lime water, tap water and other things. Record all the color changes in table 2.3. Figure 2.8, the changes in color of the red rose extract on adding lemon juice A and soap solution B. So here in test tube A, the extract remains red, while in test tube B, it changes to green. So this shows that the red rose extract reacts differently with different substances. Lemon juice and soap solution do not produce the same color in the indicator. The change from red to green in test tube B tells us that the red rose extract can help us distinguish between different types of substances. Discuss your observations with your classmates. Other samples that change the color of the flower extract to a shade of red, the same as those that change the color of blue litmus paper to red, group A, table 2.2. So, let's compare our observations. Which samples change the red rose extract to a shade of red? We find that lemon juice, amla juice, tamarind water and vinegar produced a red shade. Now think back to the litmus activity. Aren't these the same substances that change blue litmus paper to red and were placed in group A? So we can see that they are the same substances. Are the samples that change the color of the flower extract to a shade of green the same as those that change color of red litmus paper to blue? Group B, table 2.2. So now let's look at the samples that change the red rose extract to a shade of green. We find that soap solution, baking soda solution, lime water and washing powder solution produced a green color. And they are the same substances that changed red litmus paper to blue and were placed in group B. This shows that red rose extract and red litmus paper give similar results for the substances in group B. The green color of the rose extract helps us identify the same group of substances that change red litmus paper to blue. Other samples that do not change the color of the flower extract the same as those that did not change the color of red and blue litmus papers? Group C, table 2.2. So now let's look at the substances that did not change the color of the red rose extract. These include tap water, sugar solution and salt solution. They are the same substances that did not change the color of either blue or red litmus paper and were placed in group C. So, the samples that did not change the color of the flower extract are the same as those that did not affect either litmus paper. This further confirms that red rose extract behaves in a similar way to litmus and can be used as a natural indicator. From the above activity, we can conclude that the red rose extract can also be used to test the nature of the substances. Hence, it is another example of an acid-base indicator. We can conclude that the red rose extract seems to give red color in an acidic solution and green color in a basic solution. Can you now fill in the nature of the substances in table 2.3? So, after comparing the results of the red rose extract with those of litmus paper, we can see a clear pattern. The red rose extract turns red in acidic substances and green in basic substances. Therefore, red rose extract can also be used as an acid-base indicator. So, using this information, now we can identify the nature of the substances in table 2.3. So, if the extract becomes red, the substance is acidic, and if it becomes green, the substance is basic. And if there is no significant color change, the substance is neutral. We are sure that you will be excited about the above results. You may repeat the process of preparing the extract and testing substances with some vegetables, fruits or flowers such as beetroot, purple cabbage, turmeric, Indian blackberry that is jamun, and red hibiscus that is gudal flower. They can also act as acid-base indicators. So far we have used red rose extract and found that it can tell us whether a substance is acidic, basic or neutral. But red rose is not the only flower that can do this. There are many other colorful fruits and vegetables and flowers like beetroot, purple cabbage, turmeric, jamun and hibiscus. If we prepare extracts from them and test the same substances, we may observe color changes similar to those we observed with the red rose extract. This means that they too can help us identify the nature of substances through color changes. Fascinating facts. Hydrangea is a plant that grows in cooler climates in the Himalayan region and the northeastern states. It gives flowers of different colors depending on the nature of the soil. Acidic soil produces blue colored flowers, whereas in basic soil the flowers are in pink or red. Can gardeners alter the color of hydrangea flowers by adjusting the acidic or basic nature of the soil? So this is interesting. The hydrangea plant, which grows in cooler regions such as the Himalayas and the northeastern states of India, can produce flowers of different colors depending on the nature of the soil in which it grows. If the soil is acidic, the hydrangea flowers are usually blue. If the soil is basic, the flowers become pink or red. The same plant can produce different colored flowers simply because of the nature of the soil. So, if the color of the flowers depends on whether the soil is acidic or basic, then gardeners may be able to change the flower color by changing the nature of the soil. By making the soil more acidic or more basic, they can influence whether the flower appear blue, pink or red. This shows that the effects of acids and bases are not limited to laboratory experiments. They can even be seen in nature around us. So with this, we have completed the part 1 of the chapter exploring substances acidic, basic and neutral. I hope you found it easy to understand. If you want to see more videos like this, don't forget to like, share and subscribe to our channel. Thanks for watching. We will see you again in our next video. Bye bye.